Olá, boa tarde a todos.
Boa tarde, gente, a todas, todos e todes, né? Estamos muito felizes de ter todo mundo aqui com a gente. Hoje nós temos aqui a professora Andresa, né? E eu, que é a professora Cíntia, e o nosso convidado especial, que é o Dr. Aníbal.
Gente, boa tarde a todos. Então, hoje estamos aqui no nosso segundo ou terceiro, né? Se a gente contar com a aula, seminário desse semestre. O doutor Aníbal vai fazer a sua fala em inglês hoje, então a gente já tem hoje um desafio aqui maior, que é muito importante também na formação de vocês, mas não fiquem acanhados de jeito nenhum em fazerem as perguntas que normalmente vocês fazem, podem fazê-las em português, em portunhol, em espanhol ou em inglês.
Pode tentar em inglês também, né, Andressa?
Isso, façam a tentativa, mesmo que não seja correto, não tem problema, a ideia geral a gente ajuda a traduzir para vocês lá no final, tá? Vocês estão vendo que o Aníbal é muito jovem, muito receptivo, aceitou o convite feito pela Cíntia, que também é professora aqui do IB. Obrigada, Cíntia, pela tua a possibilidade de convidar o Aníbal e trazê-lo para participar nesses nossos seminários de pós-graduação.
Andresa, esse momentinho assim que a gente conversa é bom porque enquanto os alunos e as pessoas que vão assistir estão chegando. Então, a gente já está começando a aumentar de número aí. E aí, gente, uma coisa que nós temos que lembrar, né, para o pessoal da disciplina, é que logo nós vamos enviar, né, a data e o horário para a gente combinar para conversar sobre as questões avaliativas da disciplina, né, Andresa?
Já adiantando, isso, eu não quis mandar o e-mail antes para não confundir vocês quando é o seminário de hoje, mas após o final desse seminário, então eu e o professor Daniel mandaremos já um e-mail com as informações do nosso encontro, que vai ser neste mesmo bate-canal e bate-horário na semana que vem, mas a gente envia todas as informações e já com um documento do material avaliativo que vai ser nesse semestre, tá? Então, já aguardem para a gente se reunir na próxima semana nesse mesmo horário, tá?
E aí, o horário que a gente marcar, gente, não deixa de entrar um pouquinho antes para checar se as conexões estão ok, para não ter nenhum desencontro, tá bom? Então, se a gente marcar mais ou menos umas 4h30, já tenta entrar 4h20, 4h25, para não ter nenhum desencontro, porque pode ser que a gente altere a plataforma, por exemplo, usando o Teams, né, Andresa?
O Teams ou o StreamYard, eu não sei qual que a gente vai conseguir, né, Dani? Vamos organizar, tá? Mas, enfim, acho que já estamos aqui passados 5 minutos. Posso passar a palavra para a Cíntia, Dani? Você quer falar mais alguma coisa com o pessoal?
Não, só prestem bastante atenção no e-mail que vão receber para não ter desencontro, por favor. E aí, isso que nós estamos falando são para os alunos de seminários em Biologia Molecular 1, 2, 3 e 4. Lembrando que nós temos um monte de alunos, pelo menos quase 20, que não estão matriculados, mas que vão receber o e-mail e vão ser avaliados da mesma forma, tá, gente?
Exatamente. Até que essa matrícula talvez seja feita, não sei em que momento do semestre, mas para que a gente não tenha nenhum tipo de perda de conteúdo ou de falta de comunicação, é muito importante que todos estejam presentes, tá? Nessa conversa que a gente vai mandar as informações depois do seminário. Então, eu vou passar a palavra para a professora Cíntia Coelho, também do Instituto de Biologia, nossa colega. Obrigada, Cíntia, por trazer o nosso convidado, né, internacional desse seminário de hoje, e a Cíntia, então, vai fazer a apresentação e a introdução aqui do doutor Aníbal para esse seminário de hoje. Obrigada.
Obrigada a vocês pela oportunidade, né, de trazer um colaborador aqui do nosso grupo hoje. Boa tarde para todos vocês. Eu vou trocar já, começar a falar inglês, porque o seminário do Aníbal vai ser em inglês, então para a gente começar a acostumar com a língua, tá? E também para o Aníbal entender completamente o que eu estou falando, que eu vou falar a respeito dele, né? So it's a great pleasure to introduce Dr. Aníbal As Medina. And I guess my accent is different right now from the Spanish, so sorry, Aníbal, for not saying your name properly. Dr. Aníbal got his bachelor degree in biochemistry from the Catholic University of Chile in 2014. Later on, he got his master and PhD degree in molecular biology and microbiology from the same institution. Right now, Dr. Aníbal is a postdoctoral researcher at the Northwestern University in Chicago, USA, in the Department of Chemical and Biological Engineering. Dr. Aníbal and we are right now collaborating in a project to develop a diagnostic method, first for SARS-CoV-2 and now for avian influenza, and also for dengue. And we just met online. We met because Dr. Aníbal was part of 2 different groups, also developing different methods for detection of SARS-CoV-2. And now he would be presenting his presentation has a title, right? That is Innovation in Cell-Free System for Nucleic Acids and Environment Contaminant Monitoring in Latin America. Dr. Aníbal, it's a great pleasure, and the floor is yours. Thank you very much for being here with us.
Thank you so much for this amazing introduction. Thank you, Daniel and Teresa and Cynthia. It's always a pleasure to be with you guys. I hope in the future I could be in person. But as far today, it's good to be online with you guys. So I'm going to tell you a little bit of myself. As Cynthia said, I lived in Santiago in Chile. This is a nice picture of Santiago on a great day. You can see the mountains. It's a picture of the university there, and my advisor, Fernando Federici, he has a lab. It's called the Open Technologies Laboratory, where I spent my PhD there, and also a picture of my teammates. We had a lot of fun doing the PhD. And then I moved to Evanston near Chicago. I got a Pew Latin American Scholarship, which is amazing because it provides the opportunity for a Latin American to do a 2-3 year postdoc in the USA. I'm working here in Northwestern University now with Dr. Julius Lax in a laboratory called the Laboratory of RNA Design and Engineering. This is also a nice picture of Chicago and Evanston, the campus, and a picture of the whole team. So yeah, this is the nicest pictures that you can get of Chicago, of Santiago. I'm sure if you look for Brasilia, you also will have a picture like that. But the reality is a little bit different. You know, every day in our cities, doesn't matter where we are surrounded by pollution in our air, in our rivers, in our water, and also in like viruses, right? The viruses, as Cynthia was saying, avian viruses, and our interaction with pathogens comes every more frequent because of how we interact with animals, for example, and how we interact with other humans, and how we are all connected, basically. So the questions that I'm trying to study since my PhD was, how can we use open systems, open synthetic biology, to monitor nucleic acids and contaminants in our environment? And I divided this talk in 3 parts, so we can discuss at the end of each. If you have questions, feel free to ask. The first part is understanding cell-free systems, what they are. The second part is how can we use a type of cell-free system for nucleic acid detection. And the third one is how can we use another type of cell-free systems for environmental contaminants, especially with the focus on Latin America. So going to the first one, understanding cell-free systems. So cell-free systems are basically composed of 3 things. One is a machinery, the cell lysate, for example, that contains ribosomes, it contains polymerases, it contains other sort of things like tRNAs, and all the machinery necessary for doing transcription and translation in vitro. So you can have a cell growing, then open the cell, and you will have all these components in your tube. Or another way to have this is by purifying your components. You can purify individually your polymerase, your ribosomes, your tRNAs, and so on and so forth, and have a pure system. Besides that, you will need a reaction buffer that contains energy, for example, ATP, or another molecule that helps the regeneration of ATP, nucleotides for the transcription, and other components of the buffer like magnesium, potassium, sodium. We also need a DNA template. So this DNA template needs to have the right promoter, the right ribosome binding site, and you can encode whatever you want, being a protein or an RNA. And in this example that I'm showing you here, we express a green fluorescent protein, but when you see it with a UV light and with an appropriate filter, you can see the expression, and this is right after 2 hours when we put these three things together: the lysate, the buffer, and the DNA template, and you will have all this system working for you, doing transcription and then translation. And the amazing things that you can do with a cell-free system is it allows for field deployability. It means that you can go from the lab outside because it's a non-living system, so it's GMO-free. It's active after freeze drying, so meaning you have the tube, you freeze it, then you decrease the pressure, so it will release the water content from it, and it will be like a white powder that when you add water, it will restart to work. And this is amazing because all these components, polymerases, ribosomes, are still active. And it can be used for expression of proteins, for example, proteins that will be hard to produce in vivo, like toxic proteins or lethal proteins. So it can be used for manufacturing. And I know a lot of people, a lot of groups are using these systems, for example, for the developing of vaccines or drugs that you can prepare for field applications. And also other groups like myself interested on how can we use these technologies for biosensors. So preparing a reaction in the lab that you can get outside, take a real sample, and then see if you have contaminants or a virus or something like that. So this is just summary of the first part of the presentations. Cell-free systems allow for expression without a cell, very basic. 3 components: a cellular machinery, energy buffer, and DNA. You can have this system with a lysate base, which is basically open the cells and gather all the components, or by purifying individually all the components that you need. These have advantages and disadvantages. The disadvantage is that purifying every component is more expensive. The advantage is that it's a more pure and more controllable system. It doesn't have like RNases or DNases. So a lot of people is working towards a pure in vitro system, but you can do a lot of things with a lysate-based system, which is much cheaper to produce. Importantly, these systems can work after being freeze-dried, after rehydratation, and there are applications in the manufacturing and biosensing. So if no one has questions so far, I can continue with the second stage. So it's like, how can you use these systems for nucleic acid detection? And here I'm going to start talking about a lot of the work that I did in my PhD in Santiago, in Chile. So a lot of this is data from that is already published. Here I'm showing a paper from Keith Pardee in 2014, where they have a sensor that works on paper, and it's a colorimetric-based sensor. So if you have in this case, Zika virus, it will turn from yellow to purple. And it contains the following systems. It contains a transcription translation cell-free system, in this case, a purified components one, and also a type of RNA that was designed to be compatible with a fraction of the RNA from the Zika virus. I'm going to explain a little bit later what does it mean. These 2 components can be freeze-dried on a paper or a disk and distribute at 20 degrees after lyophilization. Then a second part is like a sample collection, RNA extraction. then biochemical reaction for amplification of this RNA, and then we incubate this amplification with the paper disc, and if you have the RNA to start with, it will become purple. So it's a very easy to use colorimetric. It doesn't need specialized equipment, and it's great as a point of care system. So going a little bit into the details of this TOHOL RNA sensor is basically using synthetic biology to program an RNA. It's called the switch RNA, that without the trigger RNA, it's locked in a position where it doesn't allow the interaction with the ribosome. So this sensor in this conformation doesn't allow the expression of the downstream gene right here. But when the trigger RNA interacts with this domain, it performs a rearrangement of the architecture of this structure and leaves the ribosome binding site, this blue part here, available, so the ribosome binds to this and performs translation. So basically, it's a sensor that turns on when the trigger RNA is present. And when I was studying and when I was doing my PhD, they were developing sensors like this for Zika virus, norovirus, and Ebola virus. And then when I was more or less finishing my PhD, the pandemic emerged, and then we developed also some SARS-CoV-2 TOHOL sensors using this technology. The other component that I want to say is the transcription translation cell-free system. In this paper, used a purified component. So it has all the components for transcription and translation. More than 36 individually purified components, and only few commercial suppliers, in all of them were in the Northern Hemisphere, were selling this. And it's also very expensive because it needs to be transported from, say, the US or from Canada, for example, to Chile or wherever you wanted to use it at strict minus 80 degrees. That increases the cost a lot. For example, in Chile, 100 reactions of this kit cost $4,000 at that time. I think now it's even more expensive. So in my PhD thesis, we were exploring other options, and the lysate-based cell-free system was one of the ones that we took a lot of interest in. So they work. You grow the cells, you can have different medias, then you lyse the cells. There's also different protocols for using different tools for breaking the cells, like a homogenizer, sonicator, bit beating, so on and so forth. Then you process the extract. You can do dialysis, for example, a runoff to take all of the current process to an end, and you have a cell-free extract. Then we spent a little time exploring the different energy systems. So one that was expensive, it was called 3PGA. And this molecule is expensive because it's a little bit unstable and also needs to be shipped under cold chain because it has a phosphate that is reactive. And this is why they use it for energy, because it can phosphorylate ADP, generating ATP. Another molecule that was very, very cheap is called maltodextrin, which is basically a polymer of several molecules of glucose. And we explored the use of maltodextrin. So this is a little bit of the biochemistry. 3PGA is here, 3-phosphoglycerate. It can phosphorylate ADP to generate ATP, which is the important molecule for energy. And maltodextrin is a polymer, right? Upon the interaction with phosphates, it generates glucose 1-phosphate, and then so on and so forth, the whole glycolytic pathway. So energy is important. And then this is the first figure of my paper. We compare our in-house made cell-free systems with the high-cost energy source, 3PGA, versus the maltodextrin with the phosphates, hexametaphosphates. And as you can see here, we achieve better results with our low-cost energy source. This is just following the expression of a fluorescent protein, unregulated expression, constitutive expression. And we compare this with two commercial sources. So the Pure Express one is here in purple. This is the one that they use in the sensor paper. And as you can see, all of our in-house production outperformed. They were better than this system. And we also test another one from other company. We have a little bit of variability, as you can see, if we prepare an extract on day 1, day 2, day 3, these are the number of the names of the batches, they perform a little bit different because the process itself, it's a little bit complicated. You pellet the cells in different stages, or you prepare maybe the buffer a little bit different. So we have a little bit of variation, but we can work with that because it serves our purposes, right? We can express proteins at this level. I did a little bit of the math on how much does this cost. So if you buy the Pure Express kit, the expensive transcription translation system, per reaction is like $7.8 if you buy it in Chile. And we also have some collaborators in Cambridge at that time. It was $6.5. So it's actually more expensive to buy this in Chile than in a developed country like the UK. Then the maltodextrin reduce the cost by 2 orders of magnitude. So we can actually start working with this. Then we did some other analysis, figured out what are the most costly reagents, and then we can talk about this. For example, the ribonucleotides are also a great part of the cost. But so far, we can actually work with this because our method is cheap. And the Pure Express system that was used on the paper that I showed in the beginning is prohibitively expensive. So now we can actually prepare our extracts with maltodextrin. So I did a few comparisons, comparing the performance of the Zika virus tohol, expressing an enzyme called LAC-Z, and then LAC-Z with the substrate that is yellow, it turns into red or purple later on. And I compare side by side with the commercial cell-free system called Pure Express, the expensive one, or the homemade that I made, incubating it with the corresponding trigger or with the trigger for the other sensor. So that's why we have the correct match and then a reaction that shouldn't happen. And as you can see right here, our homemade system actually is very comparable at the colorimetric level to the Pure Express, and you can see on the B the time courses of this reaction. So probably Pure Express was a little bit better, right? Because the signal and the dynamic range of the sensor, for example, for Zika virus 27 here, it has a higher dynamic range than ours. But overall, with our system, you could tell if you have or not this RNA. We study also how the different amount of DNA that you have in this system can affect the reaction. So for example, if you see Zika virus sensor 8, we call this is a little bit leaky because the untriggered reaction also has some signal, right? If you have too much of the DNA encoding for the sensor, you wouldn't be able to see the on and off difference, right? So you need to find the right amount of DNA for a sensor to have the highest dynamic range, meaning the highest difference between the on and off signal. For sensor 8, we need low amount of DNA. For sensor 27, for example, right here, we are okay with more DNA because it's less leaky. This is a feature, internal feature of the sensor, and we think it's related to how this thermodynamic structure is prone to open or to be closed. We then use a commercial amplification system for isothermal amplification of RNA. So we have RNA here at different concentrations, and we did an isothermal amplification. This system contains few enzymes. First of all, you have the RNA, a primer, then a reverse transcription makes hybrid between RNA in red and DNA in black. Then another enzyme called RNase H. It's called RNase H because it eliminates the RNA on a hybrid RNA DNA. It eliminates the red RNA, leaving space for another primer to bind. making double-stranded DNA, which is the substrate for the other enzyme, the T7 RNA polymerase, and it will make a lot of more of this RNA, and that's why it's a circular, it's an amplification method. So we tested different concentrations from 0 to low nanomolar or high nanomolar, and you can see without the amplification, so the blue light, we can see a signal only until 0.27 nanomolar, and then it's off. But when we include this amplification step, you can see 0.27 picomolar or 2.7 femtomolar, which are relevant concentrations. We didn't work with actual samples, but this is the reported concentration, for example, of the Zika virus in urine or the Zika virus in saliva and serum, right? So, yeah, we were very happy with this because we achieved sensitivity towards the limit of detection that is required, for example, for a human patient. Then, I mean, so far until here, we were using this toll sensor, which is this thermodynamic structure that recognizes another RNA from publications that were not ours. We were like, can we learn how to make a toll sensor for another RNA? So we studied the algorithm that was published by Alexander Green, and we have a version of this algorithm available on GitHub. If you want to try it, it's open over there. Basically, it takes in consideration the free energy of the structures of the switch and the interaction with the single-stranded trigger and the energy of the complex. It also considers some defects that can happen, for example, alternative structures that can happen on the structure of the toll sensor. This is the RNA in reality is not always linear like this. It can have some structure, so it considers this as well. So we studied the PVI virus. It's a virus that affects crops. It affects potatoes. It's worldwide distributed, but in Chile it's important because there are farmers of this crop in the south of Chile, and also we know in Peru. But we were in touch with the farmers in the south of Chile and with collaborators from the Faculty of Agronomy, and this is super important, and it'll be awesome if we have a sensor that works in situ. So we studied this, we studied the genome, we found some regions that were interested because they were conserved, and we designed, using our algorithm, we designed a library of possible toll sensors. Then we did linear DNA to screen in the experiment how these different sensors would perform. And we also, I'm not showing this, but we also have an E. coli strain that is engineered, so the linear DNA is more stable. And I'm showing here the 8 sensors that were the best ranked in our in silico computational designs. And what was the endpoint value when we incubated this sensor with the trigger, so with the right RNA or with another RNA.
Aníbal.
And as you can see here, yes.
Aníbal.
Yes.
And if you want, you can use the pointer that it's available in the StreamYard to show for the students.
Can you see, for example, can you see this?
So. Yes, it's perfect. Thank you.
Sounds good. Thank you for letting me know this. So yes, we have the different sensors, 1 to 8, and you can see here how was the endpoint value of the reaction after 2 hours when we incubated this sensor with the trigger or with another RNA. So you can see here, sensor 1 was very good, sensor 2, sensor 7 was also very, very good. The best one was sensor 1, and we compare here side by side sensor 1 that we designed for the PVI virus with the one that was published before for the Zika virus sensor. And as you can see, they are very specific, right? The PVI virus sensor only is on when it's incubated with the PBY RNA, and vice versa. So the Zika virus 27 sensor is only active when it's incubated with the Zika virus, but not the PBY, right? So these are sequence-specific sensors. And we think we did a very, very good job with sensor 1 and with sensor 7 that I'm showing you here. Also, when you have these at different input DNA concentrations, you achieve different dynamic range. For example, sensor 7, PBY, when you have high concentration of the input DNA, the difference between the on and off is lower than at lower concentration, for example, here. So each of them needs to be optimized, say, for the best concentration to achieve the highest dynamic range. And also, another feature that is important is the time to signal. So for example, PBY sensor 1, you can say in 1 hour if you have or not your RNA. The other important feature that is important to develop point of care, so to go outside the laboratory, is the ability to be lyophilized. So our first experiments, we had poor stability of the lyophilization. For example, we have here the Zika virus 27. We freeze dry it, and then we expect it for like 1 day or for 7 days. And then we incubate it with the trigger RNA. As you can see, after 1 day, you can see the difference, but after 7 days, it's much poorer, this difference between on and off. So we work, how can we improve this stabilization after the lyophilization, after being dry? So we did, for example, these two advantages, these two changes. One is to have special bags that you take out the air. We also add some inert gas like argon or nitrogen to the reaction, so it removes the oxygen. We think the oxygen is, is an oxidant, so it can harm the lyophilized reaction. We also protected it from the sun, and we also explore other chemicals that could be, that if we add them, it can protect this freeze drying. These are called lyoprotectants. And also we compare a professional lyophilizer like this with a desiccator, like this one. So the difference is 20 times the cost of this machine. This is a $20,000, for example, in Chile. It's a professional lyophilizer, decrease the temperature and decrease the pressure. And in this case, it's the same temperature, but without pressure, and also silica. And surprisingly, we found that the low-cost drying was better than the high-cost lyophilization when we use maltodextrin. So for some of the experiments that I'm showing, we used the desiccating system. We realized if we add lactose, we have an improvement of the signal. And these are different fluorescent proteins that were prepared, like the cell-free system was prepared from our collaborators in the UK. It was shipped to Chile. It took 3 weeks for us to have the package, rehydrate that, and run the reactions. You can see the different fluorescent proteins at white light or under UV. And an orange filter, as you can see, we have expression of everything. And in some cases, the desiccating outperform the freeze drying. That was very interesting for us, and it was published on 2022. We also tested some sensing reactions with these systems. And long story short, it works with the desiccated or the freeze dry. When we have the sensor with the right trigger, it turns the reaction purple. So we were in the middle of the pandemic when this happened, and our collaborators in Canada designed some sensors. Sorry, can you still hear me? Something changed on my audio.
Yeah,
yes?
we can hear you, yeah.
Okay. Nice. Okay. So we were in the middle of the pandemic, and our collaborators found this RNA toll sensor for the SARS-CoV-2. You can see here in 60 minutes, we can detect the RNA of the COVID virus. This started a collaboration between Keith Pardee, which is the group leader, with other collaborators in Latin America, with the group, for example, of Lindomar, in Brazil, in Santiago, with the laboratory of Fernando Federici, and so on. We have now a network of collaboration to build deployable capacity for sensing beyond now for COVID and also for beyond COVID. I have to say a special thanks for the people that is continue working on this project, especially to the postdoc Jefferson and Professor Lindomar, also Severin in Chile. They are doing an amazing job sharing all these reagents and all this capacity for our collaborators in Latin America. So yeah, summarizing this part, I know it was a lot, but that's why if you have questions, please don't hesitate to ask. So the take-home message are here. Our system was cost-effective, right? We show a method that we can detect RNA using RNA toll sensors that was cheaper because the other one was prohibitively expensive. It was we could use previously published RNA toll sensors, but we also implemented an algorithm to make novel toll sensors for the PBY virus. We compared two different lyophilization or dehydration methods, and they turned to be comparable or even better for the low-cost dehydrating systems. And there are ongoing collaborations and efforts to make these reagents and other reagents into Latin America to be a more decentralized system. Yeah, or my third topic that I wanted to talk about is monitoring contaminants using cell-free system. And this is more on what I'm working right now. I'm not able to show any data that is not published, but we work on this goal of making sensors for water use. So almost 80% of the world population is at risk of water insecurity by water scarcity or by contamination of the water. This is a lot of people, and as you can see, the more the redder this color is the highest this insecurity is, and it correlates with the more populated areas, right? And the problem that we have right now is like this: the methods that we use for detecting toxic species and for detecting, for example, metals in water are centralized and require a lot of expertise. They require scientists to run this method. So it'll be super cool if we have systems that you can actually test your water right before you use it or before you consume it. So the lab where I'm working on, the LAX lab, has some history developing sensors for water. One of the examples that I'm going to show is using the fluoride riboswitch. So riboswitches are naturally occurring sequences of RNA that can control the gene expression based on the ligand interaction, In this case, the fluoride riboswitch recognizes fluoride. So without the fluoride, this RNA polymerase will read this DNA, produce RNA, and this RNA will fold into a shape that wouldn't affect, that would terminate the transcription. It will generate something called transcription termination. It's a hairpin with a poly-U. This is a signal for the polymerase to be released from the DNA. But when fluoride is present, fluoride will, we say, it will stabilize this aptamer. It will make the RNA folds into a different structure. And so the terminator won't occur and the polymerase will keep going. In this case, you can see the effect of that transcription with a difference in color. This system was also be able to be lyophilized and rehydrated. As you can see here, the limit of detection is about 0.5 millimolar of fluoride, and is very sensitive, and it doesn't turn on when you add some other salt. People from my lab use this sensor for fluoride for detecting samples from the real world, we say, from Cartago, for example, in this case, from Costa Rica. It was near a volcano, and the samples contain fluorine, and we were able to measure that using this system. Another platform that I want to mention here is called Rosalind. Rosalind, because the meaning is RNA output sensors activated by ligand induction. It contains several layers that I'm going to talk right now. It contains, for example, a polymerase. It contains transcription factors that can sense a given molecule, and also an output that is a fluorescent RNA aptamer. This is an example of tetracycline sensors. For example, the polymerase will be able to find this DNA promoter and transcribe this RNA. It's called broccoli synaptamer, and upon the interaction with a dye, it will fluoresce. Unless we incubate this with the tetracycline repressor, this repressor will bind to the TET-O, it's an operon on the DNA, and it will prevent the polymerase to transcribe this RNA. But these sensors work, and they work in the cells too, like this. The molecule that they detect interact with the protein and release it from the DNA, and therefore the polymerase can again transcribe. You can see these are the dose responses curve. So if we don't add the tetracycline in this example, the polymerase is not able to transcribe the reporter because the transcription is blocked. But when we add the tetracycline, then we release the repression basically, and the polymerase can keep going. And we are able to visualize the expression of this RNA by measuring fluorescence. This is a very modular system because we can replace, for example, the operator with another protein. So, for example, here I'm showing the TET-R repressor that can sense tetracyclines or doxycyclines, but we can also use other transcription factor of these kinds that are repressors, for example, for sensing copper, or for sensing zinc, or for sensing small molecules, and so on and so forth. So if you change the transcription factor and the operator, you can have a range of different analytes that you can detect. What I'm going to show here, because they show a lot of analytes, but I'm interested in showing that we can actually use these technologies for in situ monitoring. This is in Chile, in my home country. You know, my home country, Chile, is very famous for the mining, for copper mining specifically. And there are a lot of places where we know there's contamination of copper. Maybe a lot other places that we don't know yet. But in this case, I'm showing these two places near mining activity that we know they contain high concentration of coppers in superficial waters, right? So they took these sensors that were developed in the lab, freeze-dried, and then used it in situ. And on C and D, I'm showing the validation using a professional technique called flame atomic absorption spectrometry. So you can calculate the exact concentration of copper in your sample by making dilutions. But also in situ, we were able to measure the copper presence using this fluorescent reaction with a Rosalind scheme. So here are the different dilutions. When it's highly concentrated, you can see it has like a toxic effect. This is because the matrix effect affects the transcription process, but there's a peak, and then all of these points are different than the one that's just hydrated with pure water. So no copper is here, and then the more copper you have, the higher the signal, and then you have some toxicity. But the key point, the message to take is we were able to use these sensors for point-of-care detection of copper in surface waters in Chile. Another example that I want to be emphatic about is in Argentina. So this is, for example, the presence of arsenic in the world, in different parts of the world. And as you can see in this part near Buenos Aires province, it has a lot of probability of having high concentration, in this case, arsenic. But we know that this region called the Matanza-Riachuelo River, it has been contaminated for a long time, for more than 200 years. And this is a place where more than around 12% of the Argentinian population lives, and it's been highly contaminated with lead and with other contaminants, being a problem for human health even. So after 2006, it was some activity from the Argentinian federal Argentinian justice system, and they realized that the state has some responsibility on this contamination. So they made this office or service called ACUMAR, which is responsible now for monitoring in several points of these rivers the presence of different contaminants. This is a collaboration. I'm not really part of this work. It's a collaboration from Dr. Diana Capdevila lab and Matias Villarruel. Both are collaborators of my projects, but this is something that they are working on. And this is, for example, a map of the region that is affected in Argentina. They are taking, the ACUMAR system is taking samples from 130 points, and they are sampling and measuring every 3 months, which is, a lot. This is how it looks in real world, and this is how it looks on the map. They are measuring pH, conductivity, temperature, but also some metals. In particularly, lead is one of the most important ones. So as I said, they're measuring 130 places every 3 months. And this is a little bit complicated to understand, but if you follow me, you won't be lost. So the different colors means different places of these 130 points. And this is a time course from June 2008 since October 2019, right? So you can see these are results from the ACUMAR office. You can see some points have like higher points of contamination, but this is only measured after like once every 3 months. So when they see this contamination, they really don't know where it's coming from. Like, is this happened yesterday or 2 months ago? They don't know. So it's ideal to have a system that allows for more continuous monitoration, that doesn't rely on expensive equipment that is centralized on labs, but something that more deployable and feel usable. That's why the Rosalind system can actually be used on a problem like this. This is an ongoing collaboration between Diana and ACUMAR, and I know they are having some stress right now because the latest development in Argentina, where the state funding is getting shorter and shorter, being this a huge problem anyways. So we look forward for this project to reach good results, but maybe I can talk to you about this later on when the project is more developed. So the take-home message from this, first of all, the water scarcity is actually a real problem. self-free systems can be used to detect contaminants in water. The Rosalind platform is modular and is deployable, and there's an urgency to frequent water monitoring and an opportunity for these systems like Rosalind to be used on the real world. And the last point that I think is important to realize is like these problems that affect us in Latin America and that affect others in other parts of the world may require local solutions. And that's why I think it's important for us to develop our own set of technologies in Latin America that we can use for our own problems. So the only thing that I can say right now is thank you for your attention. Thank you for the organizers, and special thank you for all the people that is involved in the projects, especially for my mentors, Fernand Federici, and now Julius Lux, and all the people involved in the project, especially Akumar and the group from Argentina too. Thank you.
Thank you, Aníbal, for this amazing talk. So exciting, in lots of aspects. But
thank you so much.
Very, very nice. Cynthia, do you want to start the conversation? Please, be free.
Thank you, Aníbal, for the great presentation. I actually cannot see the questions from the students, so I'll start with my own question that we actually start discussing the project that we are collaborating because of this question, right? I guess self-free systems are really good and they are a great promise, but just to start the discussion, you said several times in our talk that because we have a lot of proteins that have to work on those self-free systems, even though they are self-free systems, we have proteins to work, that needs to work. We do need to transport them, if I'm not mistaken, with freeze, right? And you don't see that as a limitation in countries like ours, that we have a lot of problems with transportation, especially frozen transportation, right? How can you see that? How can you see a solution? How this technology can be spread, which would be the limitations of, and we will have to look for other technologies, because as you said at the end of the talk, I totally agree with you, and I do believe that we have to share technologies, open share technology to have solutions for the great problems that we have, right? So I just want your opinion on that. Thank you.
Yes, thank you so much for the good comments from all of you. And yes, I think it could be still like a limitation, right? So like the proteins in general don't like when you change them from one condition to other, when you change the temperature. The self-free system is very robust, it's surprisingly robust. So thinking about all the things that are inside this transcription translation system, they tolerate a lot, right? Now, if we are thinking of something that needs to be, precise, quantitative, so on and so forth, it's better to have the conditions like super well established and don't change them too much. In any case, it's better, right, to have local production of these kind of enzymes too, or these kind of proteins too, because it's not the same, say, to produce this in Brasilia or in Santiago in Chile or in Buenos Aires and then locally deliver from an internal, like networks, than to get like a ship that is traveling for months at -20, yes, in general, the systems are robust. If you need something that is like very well defined and really precise for like some for some applications, I think it's still better to produce that locally.
Very nice. Very interesting. So Cynthia, can I do a question? And then we can go later to the questions we have here from our students. So Aníbal, I was wondering, this is a very simple question, but I think it's. I thought I'm not sure whether is able to do this or no. You can also use eukaryotic lysates to do your experiments, like for the cell-free reactions, or no? It's usually you use prokaryotic cells lysates.
Yes. So really good questions. There are a lot of cell-free transcription translation systems. The ones that are most established or higher yield, they are all from E. coli.
E. coli.
And they are also E. coli, so prokaryotic. And there are other versions from, for example, that I know of. They are the reticulocytes ones, or the wheat germ. I think that's another one. So from humans or for plants. And there are also other ones from CHO cells, for example, so from eukaryotic systems.
Okay.
So some of them still use, for example, polymerases from phages, like T7 polymerases. I'm going a little bit into the details. So having a pure system from eukaryotic, I think it'll be pretty hard because of all, like in general, it's just harder to get all the processes right. Especially, I'm thinking, for example, later on, like splicing.
Yeah,
that will be super hard to have in vitro. But there are some systems that can combine using a phage polymerase that's super efficient with some other translation system from eukaryotic that works. So they have been used, for example, for internal entry ribosome sites, for example, irises and things like this.
So Cynthia, I'm gonna do some questions from our from people, from our students. We have one from Lucas. Lucas says, Drew and Katalin won the Nobel Prize due to the development of technologies of mRNA and its delivery from the perspective of vaccine development. So does it has application in your field of research? I think it's the uridine, right? The pseudo uridine used to develop the mRNA with the pseudo uridinyl.
Yeah, we think a lot about this because that Nobel Prize is just beautiful. How the story of Katalin Karikó, how she fought for this idea for a long time, and after like trials and trials and trials, she realized this modification of the RNA made it so it can go into cells and not be degraded. We always think, like now that I'm in a more RNA lab, we think on all the other things that RNA can do. Not only transcribing, but all the shapes that it can have and all the regulations that it can have. And the modifications is one of the main ones, because you don't only have 4 nucleotides, right? You have this one, but it's modified. This one, but it's also some other type of modification. We're discovering more and more modifications, and some of them will have maybe some physiological effect. I think the RNA world is just on a good time, because a lot of people is exploring this kind of modifications and what's the biology of these modifications and how can we use this new knowledge for developing new things.
Yeah. So
for sure, super excited, but I haven't worked yet on any modification, but I'm so looking forward to try things.
Very interesting. So we have another one from Vanessa Xavier. She asks, congrats for the presentation. Just to confirm, regarding diseases caused by RNA virus, when you say sensor, is it mean this technique was used for developing medical devices for detection she said also like rapid tests. Thank you. I think, Aníbal, could you go back to the toy hold mechanism, please?
Sure.
We can.
Yes.
I think you can. We have plenty of time, so if you could explain again, if you want, if you are okay with that, and because this is
Sure.
so interesting,
Nice.
and I
Okay.
think that would be great to hear you again, because you had like a specific time, so now you can go like not that quick.
If you
Okay.
could
Okay. Yeah. Sure.
through Aníbal, among the tests, so the students could realize that even though they are self-free tests, they are actually fast tests. They do not take too long, like qPCR, like we were doing in the beginning, RT-qPCR. That would be good, I guess, for the students.
And which would be a very huge advantage compared to the RT-qPCR, for example. Yeah.
Yes. So okay. So all of this is true, right? So when you have a disease that needs to be detected by qPCR, you need to go to a centralized office where you have a nurse or someone that is actually been trained on how to take a sample and then process that sample with all the like protective equipment, extract the RNA, make the reaction. So it's costly and also time consuming, right? So like the fastest test that we had, maybe they were I don't remember exactly, but it'll be like maybe 4 hours from the sample to the result. And in this case, we're still talking. We still have some processing to do, right? We still have some process that we need to do from the RNA extraction, the isothermal amplification. Highlighted, but I think we can go faster. Sorry, I'm laser point. Okay, so for the Tuchel sensor itself, going into, okay, so sorry, one other thing that I want to say is the regulation. So you can have a sensor, for example, or a detection system, but now if you want to use it for human health and for diagnostic purposes, there are a lot of stages of validation and a lot of stages for regulation. For one of the things, for example, if you buy an RNA polymerase from a supplier, like a professional supplier, and you want to use it for research purposes, it has one cost. But if you want to use it for detection of human health markers, it has another cost. It may be the same polymerase, the same protein, but the license is different. And this is affected for the markets and stuff that I don't know well how to go around these things. But for example, when the pandemic was hit, a lot of the things that we discussed, how to use technologies like this, maybe one of the things that we thought is like maybe we are not allowed to tell the people like you have this virus for sure, because we're not able to like confirm that, but maybe we can give like a hint, or maybe you pass the screening test and now you can go to take isolation and do the real validated test. There's a lot of things that we can talk maybe with other people later with the regulation, with the process, in order to so it's ethically correct to say that you have this virus or not if you're using this technology. Like it really goes beyond the technical level. But going back into the technical level, that would be the ideal. It would be ideal to have a system that we can actually test humans, right? And tell them like, maybe you have this, or maybe we can test environmental samples, or for example, with the aviar flu, to test places where the migratory birds are. and see if they have new variants of viruses, for example. Okay, going back to the technical details, the switch itself, the mechanics of this, this is all regulated thermodynamically. So the first step is the transcription. So it will transcribe this RNA structure and it will fold. This is the predicted most frequent structure, thermodynamically speaking. It contains a domain here called the Toll domain, and then a hairpin, right? That is on the loop of this hairpin is the RBS, so the ribosome binding site right here. Because the interaction is like that, the ribosome wouldn't be able to interact with the RBS and displace this hairpin. It will need the RNA. The RNA does this, binds to the Toll domain and performs something called strand displacement. So it breaks some hydrogen bonds from here, and
the secondary structure, right?
Secondary structure. But the way it's doing this is breaking these hydrogen bonds and making new hydrogen bonds, basically. So it's destabilize this hairpin, generating another structure now that is more stable. And this makes the RBS right here available for the interaction with the ribosome.
And then the ribosome can translate.
And then the ribosome translates, yes.
Which would be a reporter gene, right? A reporter protein.
Yes.
Okay.
A reporter protein.
But my question is the sensor is actually provided by the cell-free system, right?
The sensor is provided, but we provide it in the form of DNA.
Okay.
We provide,
so, and the system starts to transcribe to produce the sensor. Okay.
Exactly. Yes.
Yeah. Mm-hmm. Very interesting.
Yes.
So and the trigger RNA that will break the secondary structure for the sensor would come from, for example, Zika virus. Zika virus would be the trigger. So in other words, if the virus RNA is present, it would make the matching with the sensor, break the secondary structure, and allow it to the ribosome to transcribe the reporter gene, right?
Yeah, the ribosome. Yeah, okay. Exactly. The trigger RNA binds to this domain. The feature of this domain here called the Toll is that it's linear. So it's meant to be not interacting with anything, just like linear part. And this is a linear interaction.
Yeah, and available to make a match with
with a portion of the trigger.
With the target. Okay, that's could be norovirus, could be Ebola virus, could be SARS-CoV, could be Zika.
Yes.
Very interesting. Yeah.
Yes.
But I guess
and then,
in this case, you also the virus also amplified by the isothermal
yes
enzyme, right? So you have a lamp to amplify the yes, that's right.
Yeah. Okay.
For example, a lamp or another type of isothermal amplification. Yes. A lamp is a little bit more complicated than NASBA, because the lamp it has so many primers, like several pairs of primers, and it will generate a DNA product, for example, that is more complicated to model. It can be done, but it's more complicated to model. But the NASBA reaction, maybe I forgot where I have it. Give me 1 second. Maybe I'll find the NASBA. Where is this NASBA thing? Here. NASBA is another kind of amplification, and is especially good for amplifying RNA. So you can have a long piece of RNA, say a full genome or a full or a full part or full transcript of an RNA virus, for example. then it's composed of the reverse transcriptase enzyme that will make RNA, DNA, cDNA, but also the complementarity to the RNA, so it will generate an hybrid. This hybrid, it's degraded by RNase H. Some reverse transcriptase have a domain that is already an RNase H, so maybe you can have these two activities in only one protein.
Okay.
Then the other primer will bind, and this primer has a tail for the T7 promoter. It will make a double-stranded DNA with containing the T7 promoter, and there is where the T7 RNA polymerase binds and makes a lot more of this RNA that enters again in the cycle. So
and can be amplified, right?
And that's how it gets amplified. Yeah, this is when when it works. Yes.
So how do you guys are sure that the RNA is available? So you have to make an extraction for the sample, right? It's not like
yes.
Then you make the extraction to release the RNA, the viral RNA, and then you mix with your cell-free system for this reaction, right?
Yeah, so far it's a two-pot. We call it a two-pot reaction because you need to have the sample purified, pass it through this RNA amplification. The advantage is that it's isothermal.
Okay.
So it doesn't require a thermocycle, for example, that will heat, then cool, then middle, then heat, then cool. Just stable 41 degrees.
Which make me think
and then you take that into the cell-free system.
Interesting, yeah. So, but it makes me think about that usually when we have an RNA virus, that there are so many secondary structures to stabilize genomes.
Yes.
So,
of course,
isothermally, it's kind of really tricky to get a linear structure to do this kind of, for example, reverse transcription, right? So that could be a limitation for your technique. Or in other words, you have to choose to pick exactly a region of the viral genome that's not on a secondary structure, right? To make sure that it's available for this kind of amplification, right?
Right. So this goes into the primer design, for example. We normally have some parameters to choose what is the best RNA. For example, if you have a virus, normally there is one RNA that is expressed, say, 10 times or 100 times more than the other RNAs of the same virus. Yeah. For example, normally for RNA negative strand virus is normally the capsid RNA. So like the RNA that encodes for the protein that will encapsulate this RNA or will protect the RNA. Then it will also encode for a polymerase or something like that, but virulent factors, so on and so forth. But mainly the main RNA presence on a virus normally is the capsid virus. So we start with that one, capsid virus, and then we screen the secondary structures. Okay. This is all theoretically and in equilibrium, and we pick regions that are candidate for having low probability of secondary structures. And then the advantage of this is that we don't need a huge portion. Only if it's, say, a couple of 100 nucleotides, 200 nucleotides, it's fine. And we can normally develop a technique, an ASVA technique for a region like that. But it could require a lot of validation. And
interesting.
It could require some. We have these parameters, like find the most express RNA, find regions that are not in secondary structures, and then validate them in the lab. Then we find maybe good 1 out of 10 that is the best, and then we perform the Toll sensor for that one. It's a whole process of developing novel sensors. Yeah, it's a lot of screening.
Yeah, this is very interesting. Yeah, so thank you so much for sharing. And clarifying first. I think we replied Vanessa already. So let's move on with our other questions. We have a question from Luana. Luana said, developing your research in the US, did you find there is a lack of consideration for the use of this kind of systems in solving problems that are too local to the global south, like the Zika virus? That's an interesting question. Yeah.
It's an interesting question, and I think a lot of well, there's some competition in this country, right? And there a lot of people is looking for the next big thing. I know, for example, I know that some people is more interested for developing tests, diagnostics tests that can be used in space, for example, than a test that could be used in Latin America or in the global south, which is, it's good because we need to, the technologies that will be used in space are also applicable for like a low resource context like our countries, right? So I think that is good. It's good that is happening. Yeah. There's a lot of momentum and there's a lot of opportunities for people like us, to learn these things. But I think the local problems require local solutions, right? So nobody's gonna come and have the right solution. Maybe it will be even harder for us, but we're already here, I think also the other part that is important is to engage with the communities. And this is something that may require some other technical capacities, like maybe like sociologists or some other expertise that will know better how the communities interact with their water, for example, or their system, if they're willing to use these technologies, how, for example, things like that.
Yeah, very nice.
Yes, but in the technical, I think there's a lot of momentum going on these technologies.
Very interesting. Cynthia, if you want, please be free to talk. I don't know. So we have more questions here in several compliments. We have like
yes,
an amazing work and presentation. Thank you. Thank you for the great lecture, Dr. Aníbal. My question is less technical, and she made the question.
That one.
Amazing. Thank you for the great lecture. And we have a last question from our students. Is when we work with RNA, a lot of care is required. Do this sensor have any defense against RNases, or does it also require a lot of care?
Great.
I was wondering something very, very close related to this question from Joves Dan, which is because we have to have so many care about RNA, and then you put a cell lysate together with the viral RNA, and then you can have a good sensor for, I don't know. So please.
Yes, we have measured the RNA decay on our extracts, and it decays really. It has we can measure it. It's some zero, or I don't remember if it's 0 or 1 order decay, so it's like enzymatic. There's a lot of RNases in our system. I think the way it works is because when we need to have the amplification of RNA before. You need to have this amplification of RNA before. So it's a lot of RNA input into this system, and so then the nucleases are like busy eating something else, so it has some time for this interaction to occur, and voila. Okay. some people is still interested in using like a more pure system that doesn't have all these RNases, where you purify all the components, and you can also add inhibitors of RNases. You can also express inhibitors of RNases in the cells to start with, so that will decrease the problem a little bit.
Degrading, yeah.
Yes. There's also other things that you could potentially do, like filter,
Okay.
things like this.
Very interesting. So this is our last question, please, Cynthia.
No, I think I would like just to thank you, Aníbal, again, just for the time that you have here. It was a great pleasure to have you here. It was a great presentation.
Yes.
Thank you for all the answers for the students. And I guess now it has already 1 hour and 20 minutes. I guess it's okay, the time. Let's get Aníbal free of here, and I thank you very much.
Yeah, thank you so much, Aníbal, for accepting this invitation from Cynthia. We were very amazed with your presentation and the details you brought today.
Yeah.
Thank you so much. Yeah, and we hope that you could visit us in Brasilia. You are invited as well. So please.
Yeah. I only would like to say that was amazing to hear you talking about your journey in science, because it's very inspiring for the students that are here. Yeah, in the first slide, you put that you are using a Pew Fellowship, that it's very important in science for young scientists. So it's easy to know why you was invited to receive this fellowship. That is so nice. And I was wondering, when you present your studying, showing the difficulties that you have to buy the suppliers, reagents, and everything. And I was wondering if this question, for example, for students in Global North, don't have this problem, so they don't have to try to solve them. And you thinking about this, so it's amazing to present and discuss these points with our students. So really, really nice. Thank you very much. Thank you, Cynthia, for invite Aníbal, and yeah, we hope you will visit in person in Brasilia in some opportunity. Thank you very much.
Thank you.
Thank you so much. It'll be for sure my pleasure and my honor to be in person with you eventually. Yeah, let's keep talking. We maybe can coordinate soon. And
yeah.
And you're totally right. Like the people that I talk to around here, we just use what is available, and we just normally think what other things we can use, and more and more and more. And there's a lot of things that we can use here, like a lot of techniques, instruments that are just available. And for me, it's also a challenge.
The cheaper one is better, right? And sometimes the
sometimes, or you know how to replace this part because you see, something. And the other one is like kind of like a black box that you don't see what's going on inside. You need a technician to come. So, but you're right. Like if you don't see the problem, why would you try to solve anything that you don't know about? So that's why the Latin American scientists, we need to work together around the problems that we have. That's the main message, and happy to keep collaborating with any of you if you want to. Okay, thank you so much for your time, for your nice words, and I wish you the best of luck.
Muchas gracias.
Muchas gracias, sí.
Obrigado.
De nada. Obrigada a vos. Muchas gracias a vos.
Gente, então é isso.
Tchau, obrigada.
Então é isso, pessoal. Vamos encerrar a transmissão. prestem atenção no e-mail que nós vamos enviar, tá bem? E obrigado pela,
Só para os estudantes
presença de todo mundo.
matriculados no
Isso.
Biologia Molecular.
Isso
Tchau,
mesmo.
gente.
Tchau.
Tchau, obrigada.